Dr. Ortiguera is currently the lead microbiologist for the TB laboratory of the Washington State Department of Health in Shoreline, Washington. He holds a degree in public health and epidemiology and has over 12 years experience working in clinical microbiology. Prior to moving to Seattle, he worked in a clinical laboratory for the Naval Health Research Center, as well as participated in numerous research projects such as vaccine effectiveness studies against various viral illnesses, clinical trials for new vaccines and rapid tests, and supported disease outbreak investigations. At the moment, like many people in Washington State Department of Health, Ryan is involved as part of the microbiology team testing patients across the state in response to the COVID-19 pandemic. So Ryan, I thank you so much for joining us.
Well, thank you for having me here. So today I'm going to give just a brief overview of the molecular methods currently being used in Mycobacterium tuberculosis testing here at the Washington State Public Health Lab. So first off is the disclosure statement. I have no financial or other relationships with any commercial interests organizations within the past 12 months. So learning objectives. So today I'm going to be covering two things. First is to list and describe the different molecular methods that are currently being used for Mycobacterium tuberculosis testing to support appropriate test choice and results interpretations. And two, just to give a brief summary of the current status of TB testing here at the Washington State Public Health Lab during the COVID-19 pandemic, and how it's going to affect the turnaround time for clinical practices. As some of you might know, that currently we are not doing any TB testing here, and all samples are being sent to Alaska Public Health Lab. And because of that, there might be a delay in the testing, the results, because of the time when we get it and from the time when we ship it out to Alaska. We typically send samples the following day, except when it's nearing the weekend, usually like between Thursday and Friday, that we cannot send samples out because of Alaska Public Health Lab are closed on the weekends. So the samples that we receive Thursday and Friday usually get shipped out the following Monday. So first off, just to give an overview of the testing that we do for active TB disease. So all clinical samples that we get here, we process through digestion and decontaminations. And from there, we do the different testing. Number one is we do an arginine O stain, which is a fluorescent stain for acid-fast bacilli. This is typically reported out within the first 24 hours after we receive the samples. The second is we do a nucleic acid amplification test, or NAT, and this is the real-time PCR test that we do in-house here. I will go more into detail with that the following slide. And this is typically reported out within 48 hours. The third is we do a culture testing where we inoculate all samples into 3 different media: the midget broth, the 7H11 plates, and the LJ slants. Typically, it takes about 2 to 4 weeks to get a positive growth. In any AFP positive, we do a DNA probe to determine the species of the mycobacterium. If we get any positive Mycobacterium tuberculosis growth, we will then set up for primary drug susceptibility testing, and this is done through midget broth. So we test for strep, INH, rifampin, ethambutol, and PZA. This takes about 2 to 3 weeks to get any results. If there is any resistance from the primary drugs, we will then set up the secondary drug testing, which is our proportion susceptibility. And this typically takes about 3 weeks from the time we set it up to get results. We incubate the plates for 3 weeks before we read the plates for any resistance. The fifth one is the molecular detection for drug resistance. This is when we when we receive requests from submitters to do any MDDR testing for any of the samples that we're currently testing. So first thing I'm going to cover is the real-time PCR. Here we used the nucleic acid amplification test, or NAT. And again, this is an in-house laboratory developed test, and this enables us to detect Mycobacterium tuberculosis complex and Mycobacterium avium complex. For MTBC, it targets for amplification the insertion sequence 6110, or the IS6110. And basically, IS6110 is an insertion element found exclusively in MTBC. For MAC, or the Mycobacterium avium complex, the target for amplification is a 16S RNA region. The approved specimen types for our nucleic acid amplification test are sputum, BAL, and any other digested or decontaminated respiratory samples. We can't test any other types of specimens, but just know that they are not a validated specimen types for that particular sample for our PCR test. Since we're sending our samples to Alaska, in Alaska they are actually performing a GeneXpert MTB RIF test. So this is a commercially available test, and again, it's a real-time PCR that targets the IS6110 for MTBC detection. It does not test for Mycobacterium avium complex, but it does test for any mutations in the RPOB genes, which is the mutations within the rifampin resistance determining region, or RRDR. The approved specimen types for GeneXperts are raw sputum or other concentrated sediments. The next thing I'm going to cover is the CDC MDDR test. So MDDR again stands for the molecular detection of drug resistance. It's a rapid and accurate detection of drug resistance in MTBC. It was offered in 2009 as molecular testing service, and it was expanded in 2012 to include the PIRA sequencing. For PIRA sequencing, they test for resistance for rifampin and isoniazid. The RPOB is the gene that they target for rifampin resistance, and INHA and CATG are for the INH resistance. If they detect any resistance to any of these two drugs, they will then set up for the complete panel testing, or the Sanger sequencing. And these sequencing tests for ethambutol, PZA, afloxacin, and injectable drugs for any of those resistance. So the next slide is just an example of the preliminary report showing the complete panel sequencing. The first three on top, the RPOB, the INHA, and the CATG, again, it tests for mutations for rifampin resistance and for isoniazid resistance. Again, if they detect any resistance from these two drugs, they will then do a complete Sanger sequencing panel. And the Sanger sequencing panel, they will test for mutations on the EMBB, which detects for ethambutol resistance, PNCA, which is a mutation for PZA resistance. The GYRA is a mutations for afloxacin resistance, and the RS, EIS, and TLYA are all mutations for the injectables. For all the samples that we submit for MDDR testing, they will do a conventional drug susceptibility testing, which is again the plate-based phenotypic growth susceptibility testing. And the final report will include this report. So it's going to do two things. One is it's going to confirm resistance based off the molecular sequencing, and it will also test for second-line drugs, the rifabutin, the ciprofloxacin, the ethionamide, and the para-aminosalicylic acid, or the PAS. Since we're sending samples to Alaska, they are not sending any requests for MDDR. They are not sending to CDC. They have a partnership with the California Department of Public Health, where any MDDR testing is currently being sent to California in Richmond, California. But the difference between the CDC MDDR testing and the California Department of Public Health, the molecular sequencing are pretty much the same. They test for mutations in the INH, the rifampin, the quinolones, and injectables. However, they do not test molecular sequencing for the ethambutol resistance. As far as the growth-based, all the drugs are pretty much the same, except they do moxifloxacin instead of afloxacin resistance. Next slide is just, so before we send out samples to CDC for MDDR testing, we have to get an approval. So this is what we request all of our submitters to send us before we could send samples to the CDC Atlanta for testing. So we ask all submitters to fill this form out as much as possible, and we will email CDC, and if they grant approval, we will attach this, and we will attach this this form along with a dash form, like an official request form for MDDR to CDC with the samples. I've put a link at the bottom here, and this is basically, it takes you to this form, the PDF form for it. Okay, the last thing I'm going to cover is the COVID-19 response here at the Washington State Public Health Lab. So since the March of 2020, we have tested over 72,000 samples processed, and over 6,300 of them have tested positive. Almost the entire microbiology team here in Shoreline have been activated to test for COVID-19, and currently we're processing about 600 samples daily. But between April to July, during the height of the pandemic, we were testing over 1,000 samples daily, and I believe the most we have tested was around 1,400 samples in one day. We are currently working on 2 different shifts, 6 to 2:30 and 2 to 10:30 schedules. As far as the future, I mean, this is the reason why the TB testing has been put on hold here in Washington, is because the resources and the people that are required to process and sample the tests, all the samples that we're receiving, it's because of this. In the future, we are currently creating a COVID laboratory. We're remodeling an entire laboratory room, and it's going to house the Cobas and the Alinity. And what these 2 machines are going to do is basically going to do all the testing that we're currently doing, like all 50+ people. These machines are basically going to handle all those testing, and this is going to allow over 1,000 samples being tested every day, as well as allowing the regular microbiology labs here in Shoreline to get back to their regular work. The COVID lab team are currently hiring on over 50 people and being trained to use these machines, as well as learning the regular PCR testing that we do here for COVID-19. I mean, aside from those, that is basically what I have.